Good afternoon. We are going ahead with the next one, material chemistry of biointerfaces. Realize some of the fundamental information regarding the crystal structure of the solid state. Now we will keep our attention on the material chemistry, particularly of the gold nanoparticle, the silver nanoparticle, how we can control the shape and structure of the nanoparticle, and how we can apply them or how we can use them for different kinds of applications. At the end, we will talk about the biointerface, but I'll explain that what is the meaning of the chemistry. This I've already explained that what is the definition of the nanoscience. So when we deal with at least one dimension that resides in the scale of nanometer, so then one dimension of any form of a matter when it resides somewhere, uh, one to so we call that object as nanoparticle. So in the nanometer scale, our naked eye cannot pinpoint or focus on single particle. Why we need this? In fact, because the reason is we have just one planet, and with a materialistic life we have. So therefore, in order to accommodate all humankind on the limited space of the, we want that the objects which we are using, so they should be much small enough with much greater facilities, and, uh, and so that we can keep them wherever we want, rather occupying a much bigger space. So nature is not going to increase the dimensions of human beings. Cannot expect that after maybe 50 or 100 years you will have all human beings of more than 7 feet. That is not going to happen. So therefore, around this size, 5 to 6 feet, and the space on this planet will also remain like this. But the devices which we are using, those devices we want, they should perform much more applications, much more facilities. Earlier, when the cell phone technology started, you are only able to phone call or to receive the phone call. The cell phone is a kind of a small computer thing. So it means that it is basically the components which are included in the cell phone, their size. You see the size of the forceps, right? And you see the component in the electronic structure. So therefore, in order to this component, you need even much smaller component to whom you can see even. So therefore, here the technology you are going to use, so that is the nanotechnology. Likewise, in the healthcare, your arteries, they are not going to be bigger in future. The size will remain the same. Earlier, cleaning the arteries from the cholesterol, you are using the drugs, but in future, obviously, probably the drugs are going to be the obsolete kind of medical help, and you will have the smart devices or the smart robot structures. So those can be introduced into bloodstream, and they slowly, simply by scratching or moving, and they can remove the cholesterol, and you don't need to take any kind of medicine. So therefore, these mechanical objects, so they can perform your all kind of medical activities. So bottom line, in order to make a smaller device, you need even much smaller components, and those much smaller components require nanotechnology. So that is the application of the nanomaterials. We will see how can we perform some of the simple simple applications for the nanomaterial in the healthcare, but at the same time it's not the glamour of the nanotechnology which I want to show it over here, but at the same time I just want to show that the nanomaterials are highly toxic and how we can avoid the toxicity of nanomaterial. Exactly the way every medicine has the side effect and how you have to avoid the side effect. Similarly, when we have the mechanical devices inserted in the human body to perform certain medical activities, so there are always the side effects, the toxicity, how we can avoid that. So nanotechnology, in fact, is not limited to the people who have the chemistry background. Nanotechnology is the technology that is being adopted by every walk of life. You will see the engineer, mechanical, material engineer, so they are also using the nanotechnology. Life sciences, chemistry, biology, they use nanotechnology. Computer science, information, electrical engineering, so they all use nanotechnology. So nanotechnology, the word nanotechnology, in fact, refers to the kind of protocols, devices, all those objects which we are going to use for the benefit of the humankind. So those objects possessing the tiny components, in order to compose those tiny components, we need a technology whom we call nanotechnology, because that technology works at the nanoscale. This is the kind of research that is going on in the United States. You will say most of the places where the amount of research is devoted to nanotechnology. University of California is much ahead in comparison to other well-known places, North Carolina, University of Illinois, New York, University of Michigan, Northwestern University. So the people over here, they are producing excellent research. University of Texas, Houston, and the Rice University. So they are in Texas. They are doing excellent research work. Rice University is over here. So I mean to say that this is not only the places in North America where the people are working in nanotechnologies, nanomaterial, but apart from this, there are other places all over the planet. In India also people are doing excellent job. They are doing very good research work, and even there are some places in North India like the National Institute of Technology, probably in Mohali, I don't know what is the correct name of that. So they are doing excellent research work. So there are several places in India as well. So I mean to say the amount of research work that is being promoted, so there are many places in the United States, obviously many people are working in that direction. This you already learned that there are two ways to produce the nanomaterial, top down and bottom up. So I have already explained that. Now, the thing which I want to explain, once we have realized the crystal structure, the unit structure, now we will try to see that how the crystal is grown into a well-defined shape. So when the nanoparticle is being grown, it is being the growth or the crystal structure or the nucleation of the nanoparticle is controlled. So you can achieve a specific shape. That shape could be a rod, bar, or a wire. It could be a cube. It could be a prism, icosahedral, or a sphere. This all depends on the application that requires that particular shape. So you cannot simply pick any shape and to use it for any kind of application. So the shape is always specific to a particular kind of application. So I'll explain how that is. In order to achieve a shape, you have to control the crystal growth. Now you know that the crystal growth is controlled by the unit cell. We define the crystal growth of particular crystal structure for the unit cell. So you may say that this is a tiny gold nanoparticle. This is a bigger gold nanoparticle. Here only 13 atoms are involved. 55, 147 or 293 atoms are involved, or any number over here in terms of percentage. But the arrangement of the unit cell is always face-centered cubic geometry. The unit cell which I have explained in the previous lecture. So that will be the unit cell no matter what is the size of the crystal. A tiny size of the crystal or a much bigger size of the crystal. So eventually when the crystal grows, there are different ways we can control the growth. And I have already shown you this slide, sorry. This is the simple synthesis of a new nanoparticle. So by using the stabilizing the reducing agent, so we can produce atom, and then we allow them to grow in the presence of surfactant stabilizing agent. And what kind of gold nanoparticle we could see? So these images are showing that if we control the crystal growth, we could have the cubes. You see the cubes? Clear cube. It is not the one unit cell. You don't need to get it confused. It's not the one. It is the shape of the crystal, right? So this crystal contains of millions or millions repeating unit cell, right? So for the gold, if this is the cube of the gold, so it means that the unit cell remains the face-centered cubic unit cell. So it could be like this, it could be like prism. As I told you, when you have to measure the dimension of the size of the particle, always keep your attention on the scale bar. So it means that the size of this cube is, one side of the cube is close to or slightly greater than the 100 nanometer. And here is the prism, then this is the hexagonal structure, and it's a roughly spherical wires, rise, this is rise, this is also rods, you can say, but these are the bars, clear bars, or the rods. Bars, yeah, the bars, the width is much greater than the rod, so we usually say this as a bar. So I mean to say that these are the different kinds of the shapes of the nanoparticle. The shape of the nanoparticle is complementary to the color. So a particular kind of a nanoparticle, shape and size of the nanoparticle, provides the color of the solution. Because when the nanoparticle, the electron cloud of the transition metal, so that interacts with the wavefront, so thereby there's the absorption of the photons, and the question is that the absorption of the energy that is being used while interacting the electron cloud with the wavefront, and where it resides, in which wavelength, in the visible region. So the wavelength in the region wherever it resides, accordingly you see the color of the solution. So as the size of the sphere of the gold nanoparticle increases, you see the color change. It's a bright red, it's slightly changing slowly to purple, and the light blue color. And now the most obvious question is that, so this color is only because of the presence of these tiny particles, those are suspended in the colloidal solution. So suppose if I have to use this for any kind of application, this suspension must be a stable suspension. If the particles are going to settle, I cannot use them. Right? So therefore, this is the condition for the application, that the suspension must be stable. Suppose if I want to use this suspension for a kind of application, it means that every time when I produce this suspension should have the size and the shape of this particle. Okay. Now the thing is that because the shape and size of the particle, which is over here, is providing me the maximum or the optimum results for a particular application. Now let's simply take the gold dust. This is gold dust that is obtained by grinding the gold. And can I simply put it in water? Can't I obtain a solution like this? Somebody who is who is a layman, who is not very clear about the colloidal stabilization, can ask you that rather than putting so much stress in the synthesis of the gold nanoparticle over here producing the colloidal suspension, you take a small piece of gold and grind it to make it a dust and simply put the dust in pure water. And why can't you have suspension like the gold nanoparticle? The problem is that you cannot do that. By grinding it, the first thing is you cannot reach to the nanometer state. You can have a larger particle, and when you have the larger particle, another thing is by grinding, you cannot control the shape of the particle. You can, somewhere you can control the size, maybe, but you cannot control the shape of the particle when you are grinding it, right? So therefore, the shape of the particle is, if somebody asks you why the shape is important, because you see that the shape is also representing the color of the solution. We will explain that, as well as the application where we need that particular kind of shape for the gold to the silver nanoparticle. So, you see now, if your sample is giving the mixture of all the shapes, that is not a good sample for a particular kind of application. You do not know how the gold nanorod is interacting or being implemented for that particular kind of application, or the wire is going to show its effect for that particular kind of application, or the assembly small nanoparticle of roughly spherical shape. If you want to really understand that how the gold nanoparticle is showing its applicability, then the shape must be uniform. That is the reason when you are producing the sample of any of the nanomaterial, so the shape of that sample particles must be consistent. So this slide I showed you previously, and here I am simply saying that, so how can you control the shape? It is all, means obvious question is now, I have shown you very different shapes. So it is much easier to understand that how I can control the shape where the gold nanoparticle can be transformed into ribbon or a wire. Right? There are other complicated shapes, but that is not easier to understand how we can control them. So, but here, if you passivate some of the crystal plane and you leave the one that can be used and transform the nucleating center into a particular direction, the growth, to direct the growth into particular direction, so that would lead to a particular growth. So as you could see that if I'm going to allow the nucleation to happen only at this crystal plane, then the growth will grow in this direction and the nucleating center will convert into nanorod or nanowire. What is the applicability for this? So you see over the applicability, we are going to, this I have already shown you, this slide as well. So we usually perform the seed growth method in order to produce the ribbons. But the bottom line question is how to use them, because here we are talking about the applications. So the nanowire which we produce for the gold or the silver, for the metallic nanowires, people are using them to cure Parkinson, Alzheimer diseases, particularly those are induced by the brain disorder, right? So still we do not have a clear kind of a medicine which could control these critical illnesses. So therefore, the other techniques based on the nanotechnology have been introduced. So if you inject some sort of a medicine, if you want, or certainly you want to give the electrical impulses to the brain, right, you can inject the sample of the nanowire into the brain over here, and this is the image, and then you can give the electrical impulses, because the metal wire is more prone for the electrical effect, and therefore it can stimulate some of the brain dead cell and it can regenerate them. So this is the idea behind that. So in this way, the transmission of the electrical signals among the brain cells can be reactivated, and particularly that is required in order to cure such kind of illnesses. Other thing is that you can have the nanowire technology to be embedded in some kind of a device, and that can produce the electric impulses, and it can be implanted in the brain. Most of these studies are performed with the animals, so I'm not sure that if now they have started with the real test for the human beings, but for the animals they have been doing it from last several years, and results are there in the form of publications. So this is the application for you may say nanowire, nanoribbon. So similarly we have the applications for the spherical nanoparticle, prismatic shaped nanoparticle, or the cubic nanoparticle. So there are plenty of applications, but I have to show you just one or two. Likewise, I have not yet come to the magnetic nanoparticle. I will be coming eventually, I think probably in the next lecture tomorrow sometime. So this slide I'll show and I'll explain in detail. So here you have the magnetic nanoparticle, the meaning for, you see this black solution, completely homogeneous solution. When you place the magnet close to this, all particles are attracted to the magnet. It means that the particle itself, themselves, behave like the tiny magnets. So when you place the magnet, so all particles are attracted toward the magnet, and you have the clear water. This is a purified solution. There are only magnetic nanoparticles suspended in water. And when I pull the magnetic nanoparticle, by using the magnet, there is a pure water over here. Can I use them? Yes, it is possible. How we can use them? Okay, they could be in any form. I'll show you. Most of the time, they are in this shapes. Those are practically, you may say, we call them. It's not roughly spherical shape, but we call them faceted nanoparticle. The nanoparticle having particular faces, crystalline faces. They could be in cubic shape or the other geometries. Rarely they are in the rod shape. So they are the rods of the iron oxide. So you can synthesize rods as well if you want, but most of the applications of the iron oxide nanoparticles are performed with roughly spherical particles like this. How can we use them? The best way is we have the pollutant in the contaminated water. We add magnetic nanoparticle, those are functionalized. So the magnetic nanoparticle entrap the pollutants. We place the magnet. In this way, the nanoparticle with the pollutants, complexed with the pollutant, can be separated out, and now you have less amount of pollutant, or you can entirely remove the pollutant. So you can purify the contaminated water. Similarly, over here you see, this is the study of a Chinese professor. So here we have a sponge, and this sponge is in fact embedded with magnetic nanoparticles. So this sponge, when we place in the contaminated water with the blue color of the dye, you may say, right, the sponge is going to absorb all dye, and then how to collect these sponge particles, because they contain iron oxide nanoparticle or magnetized. Simply you place the magnet, and collectively you can remove that, and your contaminated water is now purified. So in this way, I mean to say that there are plenty of applications you can choose. I would suggest you that in the National Institute of Technology and NIT here, some of the most important journals from American Chemical Society, elsewhere they are available. So therefore, you should have the keywords like applications of the gold nanoparticle, and you will have lists of nanoparticle publications. You don't need to fully understand the publication. I'll simply say how to become a researcher. You cannot become a researcher in overnight or fortnight. It takes years, right? So therefore, what yes, is the right path to begin with. The right path to start with researching is, so the field in which you are interested, whatsoever is gold nanoparticle, silver nanoparticle, iron oxide nanoparticle, first simply pull down the related publications, take the printout. You may read them on the computer, but I feel that when you have the hard copy in front of you, you will have a better grasp of understanding rather than simply reading on the screen, right? So read them. So don't expect that you are going to understand everything. It never happens, right? Because this is not your work, somebody else's work. So therefore, there is no way you are going to grasp anything. You may expect that probably I'm not a good scientist, or I may not be having the appropriate understanding because I can't understand. That is not the thing. You cannot understand everything, right? So therefore, you should accept that only the part which you can understand. So whatsoever is, even if a tiny paragraph, that is enough in the beginning. Likewise, you should download the publication related to application, maybe application of cleaning the contaminated water. There are plenty of publications related to the cleaning of contaminated water by the magnetic nanoparticle. And you will see there are wonderful ways to clean the contaminated water by using the magnetic nanoparticle. And you can also download and see the applications, the medical applications extended by the gold nanoparticle in the in curing the critical illnesses. So that also you can read, and you will be able to understand how the nanoparticles are being used in a particular direction. So that is the way you start reading and putting and generating your interest in a particular direction and start thinking in that direction. Slowly you start working in that direction, and slowly it will help you to understand each and everything. That is the way one can become a scientist. Going ahead now, so there is another most important thing is target drug release. Actually, this application is very common, and you might be reading it frequently, right? And how it happens, and how how the nanoparticles are being used for this kind of treatment. You know, when you go to, I would say, what we call non-conventional doctor, a desi doctor who is not equipped with full degree, MBBS or the specialization. So he or she will say, okay, if you have a problem, take milk with the haldi and all those things, and everything is going to be all right. So no doubt haldi is excellent way of treating several things, but one thing you have to keep in your mind: if I have pure water, glass of water, and if I try to dissolve the haldi, I won't be able to do it. If I cannot dissolve haldi in pure water, how could you expect that I can dissolve it when I can drink it, right? So the bioavailability, it means the amount of the effectiveness of the molecules which is going to treat a particular illness, that is going to happen only when you have the maximum bioavailability. I mean to say that if the bioavailability is higher, only then that problem is going to be cured. Otherwise, it's a problem. So therefore, by drinking any amount of haldi in the form of warm water in the milk or anything, that is not going to help you unless it is bioavailable for a particular problem, right? So it is this happens for maximum number of medicines, because most of the medicines which you are taking every day, they are the organic molecules with with very tiny ability to be solubilized in the aqueous phase, right? So therefore most of that medicine is simply drained out of the body. Very tiny amount is going to be used for a particular kind of illness. That problem is being solved when we take the help of the nanoparticle. So therefore what the nanoparticles are going to do, nanoparticles are usually modified particles. So as I told you here, the colloidal stabilization is in action. So this is a nanoparticle, right? And now this nanoparticle is having colloidal stabilization provided by the colloidal stabilizer in the form of a shell, which is a bilayer, right? So now this particle is a colloidal stabilized particle. So this shell which is there around the nanoparticle is in fact the place where we can put the drug molecules. The drug molecules can be solubilized over here, and nanoparticle works as a carrier. So nanoparticle is taking all the drugs to a place, and it is going to release them. So I mean to say still nanoparticle, right? And the shell, colloidal shell, that is being provided by the colloidal particle to provide the colloidal stability. So that is there. So now at the target site, the drug molecules are released. Drug molecules are released. Now you see that when the nanoparticle is going to release the drug at target, that is highly effective. It is exactly the same way, rather than drinking the milk with haldi. If you simply sprinkle the haldi on the wound, that would be much more effective. Now for the external wound there is no problem, but for the internal wound, how that can happen? This is another way, right? So this is the way we treat the tumor, cancerous tumor, right? So you may have the anti-cancerous nanoparticle, or you may have the nanoparticles carrying the anti-cancer drug and simply injecting the nanoparticle over there, and nanoparticle releasing them, and probably it will help. So this is another way of putting the nanoparticle for the biomedical applications. So the main topic of this chapter is the material chemistry at the biointerfaces. We are coming to that point again. But now, after going through different applications that I've shown you over here, right? So the application here, so this was the first one we tried to explain for the shape of the molecule. This was the second, to clean the contaminated water. This is the third one, right? So again, here you need the shape. So I have chosen the spherical particle. You may ask me, why can't I choose the wires of the river? I believe carrying the maximum amount of drug by the spherical particle, that is the most appropriate vehicle that can help me, rather than taking the wires of the drugs, right? So small spherical nanoparticle, they are going to carry a much bigger load in comparison to if I have a sample of the rods of the rivers. So for a particular kind of application. So this was the glamour of nanotechnology in all these applications. As I told you that every medicine has a side effect. The same way nanotechnology and so that this kind of devices or the techniques, so they are also the particles, so they have also the side effect in terms of toxicity. So here we are talking about nanotoxicity. Let me put the drug-loaded nanoparticle and inject in the bloodstream so that they can reach the target where the drug is going to be released. But the problem is the carrying medium is the bloodstream, and the bloodstream is not only the aqueous solution as the salt solution, but it has so many other components like the blood cell, which is a very important component for the survival, right? So now, suppose if I say that these are the nanoparticles or these are the particles carrying the drug. When they are injected in the bloodstream, that is systemic circulation, these nanoparticles are not expected to break the blood cell. They are not expected to break the blood cell. If on the way they are breaking the blood cell, before reaching the target, it means they are inducing the toxicity. So you are not supposed to use such kind of carrying object to deliver the medicine, right? So you need to find out the way that the nanoparticle which you are choosing should not interact with the blood cell membrane. You should keep in your mind that any metallic surface, nanometallic surface, has very high potential to interact with the cell wall. Living cell wall that possesses very high potential. So therefore, the free surface of the nanoparticle has to be passivated so that it should not directly come in contact with the cell wall, right? So you see these are the coated the nanoparticle. Exactly, this is the coat of the colloidal stabilization. So you should choose the colloidal stabilization by that kind of molecules. Those molecules should not allow the metal surface to interact with the biological surface of the cell wall. And in this way, that is the correct way of using the application. So to avoid the nanotoxicity. So this is the nanotoxicity so the nanoparticle, if they are, it's not only the bloodstream, because bloodstream is everywhere. The bloodstream is everywhere, no matter if you are going to introduce this in the tumor. So the blood vessels are everywhere, right? So therefore, I mean to say that in order to avoid the toxicity or inducing the hemolysis, this is known as hemolysis. When we break the blood cell, the process is known as hemolysis. So the main objective for the nanoparticle that is going to be used for biomedical application, so the hemolysis must be avoided. So you could see over here, these in this image, the nanoparticles are attached to the blood cell, but they are not breaking the blood cell, right? So I also showed you the magnetic nanoparticle for my research. When I use them, so I use specific kind of the sample that is not going to break the blood cell. So this is one example of the nanotoxicity, so you should avoid that. Next one. So this is the nanotoxicity in environment. This is known as the zebrafish. Biology people know it. Very tiny fish, just probably a few centimeters long, and most of it is semi-transparent, right? So this is the normal growth. But if the water in which this zebrafish grows, a contaminated water with the nanoparticle, the growth is not normal. So you could see this is the abnormal growth of the zebrafish in the presence of nanoparticle. So the zebrafish takes up the nanoparticle. I'm not showing those images here. Because the fish, you see now, there's a very common problem of microplastic. So recent studies are saying you are already eating microplastic, particularly the fish you are eating every day. You may think that that is a freshwater fish. No doubt that is a freshwater fish. But along with the fish, you are eating microplastic. How? Because the fish is not possessing the intelligent mind to differentiate while eating if it's a microplastic or say any other micro living object, right? So therefore, fish is eating everything, right? So it is eating microplastic as well. So once the microplastic is going, that is accumulating inside the fish, and eventually you are catching that fish and eating, and hence you are eating the microplastic as well. So here is the problem. So therefore, our fresh water must be avoid from, or must be cleaned in terms of the contaminant, because eventually whatsoever is in the fresh water, that is coming on your food table. So this is the clear example that is not the eating of the contaminant or the nanoparticle by the organisms living in the fresh water or marine water. This is even beyond that, right? So when that is contaminated, the fresh water or the marine water is contaminated, even the growth of the fish is going to be affected. So there is abnormal growth, and the fin fold is straight over here, but it's curved here. There's a cardiac, so cardiac, yolks, sac, and eye, all kinds of organs are showing the deformation with respect to the exposure of the nanoparticle. So therefore, contamination, any kind of nanotoxicity that is existing in the fresh water, eventually affecting the aquatic life. So, what if we have talked about the aquatic life in water? So, what is happening outside with the plant growth? How the plant growth is affected? Again, if your soil is contaminated with nanoparticles, okay, before going ahead, you might be wondering why the nanoparticles are going to end up in the soil. There are plenty of ways. So now all steel industry, paint industry that is related to the that depends on the metallic paints with small kind of metal particles, you may say, or the mining industry where you have the effluent mining wastewater that contains plenty of those small metallic particles, right? So those small particles eventually they are ending up in the landfill or on the soil or contaminating the freshwater reservoir. So this particular plant growth is the plant growth is being facilitated by biosynergism. So these nodules, which are in fact facilitating the plant growth, they are being occupied by the bacteria. So they help in the symbiosis, right? What happens? That bacteria grows in soil, but because of the metal particles' ability to interact with the cell wall, so you see these dots. So these dots are the particles which are there as contaminant in the soil. So they are being picked up by the bacterial cell wall, and hence the bacterial growth is affected over here. Thereby, the growth of nodules, so that is affected. Eventually, that is facilitating the nitrogen fixation for the plant growth that is being affected. So, bottom line, the overall growth, suppose if you see this graph, This is the control. This is the control of overall growth of the plant in a non-contaminated soil. But when it is contaminated with the different kinds of carbon nanoparticle, so all these are the carbon nanotube or carbon nanoparticle of different shapes, you could see in all the cases the growth is much retarded in comparison to the control. So you may have the nanoparticle toxicity in water, on soil, or in the bloodstream. There are plenty of implications, so you cannot avoid the nanotoxicity, so therefore you have to find out the way how to passivate the nanotoxicity when you are looking for a particular application. Now, at biointerfaces, you might have seen that I showed this slide, I think yesterday's lecture, when we were talking about regarding the biosurfactants. So, here, how the contaminants in the form of a nanoparticle, they are going to affect your respiratory system. That is also a very prominent example, particularly for the developing country people and particularly in India. When the suspended particle concentration increases in all metropolitan city, in Delhi or other places, and you frequently read the news that the breathing air quality is very, very, very dangerous, and the people should take precautions. In fact, what you are breathing, what are the contaminants? Then the suspended particle contaminant. Most of them are the carbon fiber, because when this fossil fuel burns, and therefore the smoke which you are seeing over there, so those particles remain suspended in the air because they are being entrapped by the moisture and all those things, and hence you are breathing them. So apart from that, there are dust particle and all those things. So you may not see them. You see the haze. Most of the time the haze you are seeing, that is being created by the pollutant. It's not a natural process. The mist is the natural process, but the haze most of the time that is being created, that is because of the excessive pollutants or the dust particle in the air combining with that of the moisture, right? And you are breathing them. What they are doing eventually, they are going into the alveolus. These are the particles which are entering into the alveolus along with the inhaled air, where exchange of air is taking place, right? But these particles, what they are doing, they are interacting with the bilayer. So, as you could see that, if this particle has the ability that those amphiphilic molecules, so they can arrange over here and coat them. Exactly, there are plenty of amphiphilic molecules. So this bilayer is made up of amphiphilic molecules, those are known as lipids, right? So when the particle is going to hit this interface, and it can instantaneously grab the amphiphilic molecules. Now there is a constant supply of amphiphilic molecules from the lamellar bodies over there. But when the rate of particle those are hitting the interface exceeds the rate with which the amphiphilic molecules are synthesized by the body, so then that creates a respiratory problem. So it means that this biofilm that is being created by the amphiphilic molecules cannot be regenerated at that space, at that speed by which you are receiving the contaminant, right? So that is the problematic. So I mean to say that the respiratory problem are not alike. As I told you that we have the respiratory problem because of the side effects of the vaccine, COVID vaccine. But what are the respiratory problems in this part of the planet? It is not probably the COVID vaccine that is creating the respiratory problem, but it is the pollution, right? So therefore, this is another form of nanotoxicity. So nanotoxicity working at biointerfaces. So this is the biointerface where the nanotoxicity is working. So it means that what is the solution? Suppose if you ask me what is the solution, you cannot change the amount of the surfactant produced by the human body. It is the same for everybody, right? But you have to control the contaminants, those are being inhaled into your respiratory system. So that is the only way to avoid the pollution. So if you have the clean air, so you can have a healthy lung and healthy breathing system. But if you have contaminated air at some point, you will be having respiratory problem. At the end, nanotechnology is a scientific wonder of modern time, but it comes with an implication. You say that I've shown you the applications, but I've also shown you the implications, right? So it comes at a cost, no doubt. No doubt it's a much better way of taking care of the human health. But if you have to take care of the human health by using nanotechnology, you have to be very clear that the nanotoxicity must be avoided. Prevention is the best health cure. Nanoscopic detection is the key to prevention. So I mean to say that because the North American medical system works on the prevention, right? So there we are always under stress. We are being asked every time after a certain amount of period that this test is due for you. You have to go through this test even if you are completely healthy. Here, when we are completely healthy, we don't want to see the face of a doctor, but that is not the case in North America. You have to go to the doctor to go through that particular test because government want that before you tell the government that I am in a critical stage now, and so all the resources the government is going to put to save your life. Before that happens, government want that there should be way to detect it, that problem is now coming for you, and there are ways to prevent that problem. So this is the way. So what I have explained today in just a few minutes, we actually our first lecture was on the crystalline phase, crystalline solid state, and the reason for that because the nanoparticle examples which we have taken today, or which we will be taking in the next few lectures, so they all belong to the solid state of crystals. So that's why our focus was that what is the fundamental of crystal structure and how we can understand and differentiate the crystal from that of amorphous. And then it is the crystal that grows in specific shape. In this lecture, so we have placed our attention on the different shapes of the crystal, the shapes of the particle that is produced from by controlling the crystal growth, right? So I mean to say that when you have to use a particular sample of the nanoparticle, so that must possess a same shape of the sample and the same size. Only then you can achieve the results. Otherwise, you won't be having the reproducibility, because the one sample is consisting of maybe more nanorods, nanoribbon, and few nanoparticles. The second one could be otherwise having more number of the spherical particle and less amount of the nanorod and the nanoribbon, and you won't be having the reproducibility. And in this way, you cannot quantify the results. So therefore, in nanotechnology, the quantification of the result is related to the same shape and size of the nanoparticle sample. So therefore, the crystal growth is very, very important. When you have to have the nanoparticle sample, you should have an appropriate methodology available to control the crystal growth. That is much required. Thank you so much for your attention.
Of the nanoparticles, they have a very important role in the properties of nanoparticles. So you have told that the direction of the growth can be controlled by blocking the sides.
Yeah,
that
yes, absolutely.
affect planes. So is there any specific mechanism, requirements or conditions that we can control the direction?
Yes. If you know that which direction you have to control and which morphology you have to achieve, obviously by blocking that crystal planes, you can divert the crystal growth in that direction. So you will see that when you have to, particularly for the rod and the wire, uh, uh, it is you have to direct the growth on 110 plane. All other planes must be blocked, right? So you have to find out the amphiphilic molecules which can be your molecules are having uniform absorption. Some will prefer 100 crystal plane, some will prefer 110 crystal plane, and some on 111 crystal plane. So I mean to say that it is all uh, 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 the kind of understanding you need to require, that what kind of amphiphilic molecule those can be absorbed on particular crystal plane, so that you can have a controlled growth. So for the gold nanoparticle, most of the research that is being uh, done in this areas, so that is for the gold nanoparticle. It's much easier for the gold nanoparticle to control the crystal growth. And there are many good review articles where they have used many different kinds of amphiphilic molecules, or sometimes even the counter ions for a salt, like bromide, chloride, and iodide. So they can also help in the in crystal growth. So it all depends that what are your requirements and in which morphology you want to use. But suppose if you ask me that there is a specified methodology available, I would say no, there is not specified methodology, unfortunately. Yeah, you have to optimize the condition according to your requirement. Any other question?
Flower shaped or a plate shaped nanoparticles. They're similar or they just give us something? Yes.
Depends what I prefer to give the name of the particle, right? The most of the time, yeah, flower shape or the prismatic shape or all those. So they are the conventional way of just naming them. So whatsoever the kind of shape you are getting, so you will see what is the common name that can be provided. So everybody can understand that. So that is why the flower shape means to come from that direction. Yeah. What you see, and you try to mimic that image with some of the common image, right? So you will try to give the name accordingly. The flower-like structures, the roughly spherical particle. The word when we use the roughly spherical, you can have rarely entirely a spherical part, but there are very good publication where the people have produced a very precise sphere shapes. In most of the cases, you will see not the precise spheres, but rather roughly spherical, where you cannot make out that it's exactly sphere or some other kind of a shape is originating. So that is why in most of the cases we use the term roughly spherical. Yeah.
Nanoparticles.
Yeah.
How can we optimize the binding capacity of nanoparticles with drugs? Because all the phenomena depends upon the binding capacity of drug and
Binding capacity or drug loading capacity.
Yes.
You want to? Yes, that's a very important question. That is why when uh, you want that this particular kind of a drug you want to release at the target, you need to know, or you need to produce the environment that is the favorable environment for the solubilization of the drug. But this is not the required case. So that could be favorable under a different experimental condition. But when the nanoparticle is going to travel loaded with the drug, then there must be a environment where the drug is going to be released. So I mean to say that the one environment where the binding affinity of the drug is enhanced, so you can load the drug. And so you allow the nanoparticle to travel to a target site, and then you have to create a condition that would allow the minimization of the forces between the molecules which are creating the colloidal stability and the drug molecules. So the drug molecules can be released here. So usually the people do it different kind of experiment. The pH change. So if you have the different pH, that would allow the drug molecule. Sometimes temperature variation that can also facilitate that. So I don't know. Apart from the temperature, there could be other change the environment nanoparticle to release the drug. Okay, thank you so much.
Thank you, sir. The next tutorial session will be conducted by Dr. Jaspreet Kaur Rajput on synthesis, characterization and electronic properties of gold and silver nanoparticles in the Department of Chemistry at 2 p.m. Thank you.